All right, so this is the first set of notes for biochemistry. So this is like the first official UW Oshkosh required unit for CAP Bio. What we are going to be talking about in biochemistry is the study of compounds that contain carbon. That's literally what the word organic means, is it means it contains carbon. Carbon is really, really unique, and it's an interesting molecule or atom, I'm sorry, because it has 4 valence electrons. So from chemistry, you might remember that that means on its outer shell, it has 4 electrons and it needs 4 more in order to make a full outer ring. This is done through covalent bonding. And just like in the background in this picture where everything is like in groups of 4, like that, carbon does that too when it binds to other things. And this makes a lot of different 3D shapes. So when we're talking about the shapes that carbon can make, is it can make a lot of really, really unique things. And we draw these out in skeletons. And you guys are going to be doing a little worksheet about these skeletons in class. So for example, A is length. So we can make big, long chains of these carbon molecules like ethane, propane, butane, methane, um, heptane, octane. All of those just mean that one more carbon with hydrogens all around it was added to the chain. For B, we can also make branching molecules. So butane and then next to it isobutane, those are what we call isomers of each other. So they have the exact same chemical formula, but because they're different shapes, they have different properties, which is very cool. In C, we can also make single bonds or double bonds. So here we have 1 butene and 2 butene. So 1 butene means that the number 1 to number 2 carbon, that's where the double bond is. And in 2 butene, that means that the 2 to 3 carbon has a double bond. We can also make rings, and these are going to be really, really common when we start talking about nucleic acids and when we talk about different carbohydrates. Everything there is in a ring, kind of like Greta's little ball thing that she plays with. So you think of like these different ring shapes and all of the different shapes that those rings can make. So 2 examples of those are cyclohexane and benzene. We are actually going to skip over isomers. So hang on just a second. Never mind. I looked through my notes. We're not going to skip over isomers. So we have 3 different types of isomers: structural, geometric, and enantiomers. And these are really, really cool things. So we have isomers, and these are compounds that we kind of talked about before, that they have the same molecular formula, but they have different structures. And because they have different structures, they have different properties. So the 3 are very, very important in biology specifically. So here are just some pictures of what these look like. So a structural isomer, that has a completely different 3D shape to it. So that one on the top, straight line versus that T shape. A geometric isomer is basically a twisting, okay? So that double bond in the middle isn't very twisty. It cannot be bent very easily. So if there are 2 atoms that are up here, and then now that one gets twisted down there, it can change the whole molecule, which is kind of crazy. And then down below, we have enantiomers. And these are, the hands there are because these are handed molecules. So they are a left hand and a right hand. So just like both of your hands are the same, but they're a mirror image of each other. So an enantiomer is a mirror image molecule. And these are denoted on molecules as being an L or a D molecule. Okay. And we're going to talk about a really weird example of that. So with our structural isomers, they differ in the covalent arrangements. They differ sometimes in the location of their double bonds. And as these carbon skeletons get bigger and bigger, the possible isomers also get bigger. Stop wiggling. Okay. So we also have our geometric isomers. So down below we have those different shapes. So same formula, but they differ in their spatial arrangements. And this is due to the inflexibility of those double bonds. And this can affect biological activity as well. All right. And then lastly, enantiomers. These are my favorite molecules that are mirror images of each other. So they are considered right-handed or left-handed versions. The really unique thing about these is that a lot of proteins actually have enantiomers of each other, and the L form or the D form, which is how we denote the right-handed and the left-handed form, your body either can or can't break down. So next time you are at Walgreens or CVS or a grocery store or something like that, I want you to go in like the aisle where they have all the vitamins and like supplements and stuff like that. And if you look at the bottles, sometimes they'll say like L-carnitine. That's a supplement you can take. And that's because it is the right-handed version of that molecule. Or D something, and that's because it's the left-handed version of that molecule. This handedness is also called chirality. The D stands for demon, which is the left-handed version, and that goes back to old Catholic schools where if you were left-handed, they thought that was a sign of the devil, and then they would beat you until you used your right hand. So that's exciting. That's a fun thing. A little fun fact for you guys. Another little fun fact for you guys here. So we have two different drugs down below, L-DOPA and D-DOPA. One is the drug that treats Parkinson's disease, and the other one doesn't do anything, just goes right through your body. Kind of crazy. There is another one that is a really, really famous example called thalidomide. This was actually given to pregnant women. I'm going to be wrong if I say dates. 50s and 60s, I want to say, for the treatment of morning sickness. However, there were several labs that they did their production incorrectly. And when that happened, they made the mirror image of that thalidomide drug, and it created a massive, massive birth defects. So I'm just going to exit out of this, and I'm going to show you guys some pictures of the birth defects that this caused. All right, so if you just search for like thalidomide babies, this caused, they called them like flipper fins and flipper arms. So it caused babies to have no limb growth and stuff like that. There's an actor that I believe was on the freak show season of American Horror Story that was actually a thalidomide baby. So these are really, really common defects in these children from their moms taking thalidomide, which was supposed to treat morning sickness. So a very, very unfortunate side effect of this enantiomer. Here's a meme. If you get it, that means you understand. Moving on. All right, so the next part of this intro to biochem is discussing different functional groups. So what a functional group is it is a predictable arrangement of molecules or atoms that make a group that has a very distinct property. This is going to be really important when we talk about cells and when we talk about cell signaling, which is how cells actually talk to each other. So what these functional groups are, if we look at those skeletal drawings of different atoms, we will see some of these different groups and they make molecules, like I said, with a very predictable property. So, yeah. Oh, are you saying the first one? Are you saying, oh? So the first one is a hydroxyl group, and that is what makes up an alcohol. So an alcohol is going to be a polar molecule, and this makes the molecule soluble in water. Okay, so the next one is a C double bonded to an O. That would be a carbonyl. And these are things like aldehydes and ketones. These are also a polar molecule. What polar means is that those molecules are attracted to water. So they have like a magnetism to them. Anything that's like an aldehyde or a ketone, that is going to be a byproduct of digesting a sugar. So the next one is a carboxyl, and this is a COOH. And I have pictures of all these things. This is going to make something an acid, like carboxylic acid. And this dissociates in water, making an acidic solution. So we also have an amino group. So I'm sure you've heard of an amino acid at this point. Oh my gosh, hang on. All right, so the amino, that makes up an amino acid, which is part of a protein. The acid part of an amino acid is actually that carboxylic acid. So they're named an amino acid because of those two functional groups. Next, we have a sulfohydryl. This is what makes like a 3D structure of a protein. And these bonds are the reason why if you burn hair, it smells really, really bad because there's sulfur in your keratin protein that make up your hair. Phosphates, these make up ATP. And then next we have methyl. Don't do drugs, kids. These are nonpolar, so these don't mix with water, and they are hydrophobic, so they are scared of water. Okay, so here I have some pictures of what these different things look like, and these are all in the PowerPoint and the notes. So anything with this OH at the end, that is going to be an alcohol. So here we have a carbonyl, so a C double bonded to an O, and that is going to be a specific type called an aldehyde. When it's in the middle of a chain, that is called a ketone. And next, that's COOH. If we have a C double bonded to an O with an OH also attached to that same C, that is going to be our carboxyl. So this acid right here is actually acetic acid, and this is what makes vinegar acidic. So on the worksheet that you guys are going to get when you are circling all of these functional groups, if you see a C double bonded to an O, it's not necessarily a ketone or an aldehyde. You have to check to see if there's an OH on it as well, because if all of those things are together, then it is going to be a carboxyl group. And then here we have our amino group. So we have an N double bond to an H, and look over here, we have that carboxyl group. So that's our acid. So that's why we call this an amino acid over there. So kind of cool. We've got our sulfohydryl, so an SH. We have our phosphate group, so P with 4 O's around it. That is going to be an energy transfer molecule when we start talking about ATP. When we are looking at these molecules as well, we have a couple different terms. So a monomer is going to be like the smallest little building block. Poly means many, so that's going to be like one chain. And a macromolecule is 2 or more of these polymers that are bonded together to make a giant molecule. In this unit as well, we're just going to be mentioning how some of these different molecules are built up and broken down. So if we're going from a monomer, so one unit, to a polymer, so we are building, we call this dehydration synthesis. So synthesis means to make. Dehydration means to lose water. So, what happens in this reaction is water comes out in order to build this molecule. On the other hand, the breakdown of polymers is called hydrolysis. Hydro means water, lysis means to break. So, a polymer is going to go to a monomer by adding water breaking. Okay, so water is going to go in, and then that molecule is going to break apart. So here is just like a little picture of that happening. So up here, if we have a short polymer and an unlinked monomer, and here we have an OH group, so we have an alcohol group and an H group, those are going to bind together and leave to make water, and that connects our polymer together. The opposite is what happens in hydrolysis. So if we have our long polymer, when we add water, it gets attracted to those two bonding sites. The H gets added here, and we have an OH, so it makes this molecule an alcohol, and then that molecule will actually separate. So the four basic molecules that we're going to be talking about are carbs, lipids, proteins, and nucleic acids. And I do have separate video lectures for all of those. So today you guys are just going to focus on looking at those functional groups and basic structures of molecules before we get into specifics on each one of these molecules. So I hope I did not put you to sleep like I put Greta to sleep, because this really is some exciting stuff.